This method allows cellular structure and function to be perturbed through the fusion of two different cell types, to address questions such as how do cells respond to changes in organelle number. By imaging fluorescently tagged proteins within fused cells, cellular structures can be referenced back to their cell type of origin to study the mechanisms of organelle homeostasis and function.
For fluorescent dye labeling, seed the human cancer cell populations of interest such that each culture will have expanded to at least 6 × 10⁶ cells at a 70 to 90% confluency by the next morning. On the day of labeling, wash the root-leaden E-GFP and root-leaden M scarlet cells 2 times with 10 milliliters of PBS per wash. Dilute violet cell dye to 500 nanomolar in PBS, and far red cell dye to 200 nanomolar in PBS. Label the root-leaden E-GFP cells with the diluted violet cell dye, and the root-leaden M scarlet cells with the diluted far red cell dye for 1 minute at room temperature. At the end of the incubations, stop the reactions with 10 milliliters of DMEM for 5 minutes. Wash both labeled cell cultures and one unlabeled parental cell culture with 10 milliliters of fresh PBS before incubating all of the cultures with 1 milliliter of pre-warmed trypsin. At the end of the incubation, transfer the detached cell solutions to individual 15 milliliter conical tubes for centrifugation, and gently resuspend the dye-labeled pellets in 1 milliliter of PBS and the unlabeled cells in 1 milliliter of DMEM. Then, return all of the cell samples to the cell culture incubator.